Hello everyone. Our next module is about mechanism of blood, clotting and blood groups. So these are the contents. First there will be introduction. Then comes what is hemostasis. Then the mechanism of hemostasis. Then comes the clotting factors. Then the physiology of the clotting mechanism, blood groups and erythroblastosis fetalis. So first, as we are talking about the blood clotting and the blood groups, first we need to understand what is blood, right? So blood is a fluid connective tissue that consists what the plasma, blood cells like RBCs, WBCs, platelets, and it circulates what throughout the body for the carrying out of oxygen, nutrients, proteins to various cells and the organs. It makes up to 8% of our body weight. So the blood actually makes the volume of up to 8% of our body weight. An average adult actually carries what 5 to 6 liters of the blood. First is the red blood cells, that is erythrocytes. Red blood cells are biconcave in nature without nucleus. They are anucleated in nature in humans, also known as erythrocytes. They are rich in iron protein, that is hemoglobin. This hemoglobin actually carries out what? Oxygen. So, and it gives what? This iron part, which is present in the hemoglobin, actually contributes the color, the red color. RBCs are the most copious blood cells produced in the bone marrows. So the RBC's productions are mainly occur where? In the bone marrows, in the red bone marrow part. So their main function is to what? To carry out the oxygen from one place to the another place, that is from one tissue to the another organs. Then comes the white blood cells. So the white blood cells also known as what? Leukocytes. So these leukocytes are the colorless blood cells, which is also known as the defensive cells or the immune cells of our body. So they are colorless. Why? Because they are devoid of hemoglobin. And they are further classified as granulocytes and the agranulocytes. So WBCs mainly contribute what? The immune system, strengthening of the immune system and the defense mechanism against any foreign particles or the foreign agents. So in the granulocytes, it is consist of what? The monocyte, neutrophils, eosinophils, and the basophils. And in the agranulocyte, it is having the lymphocytes and the monocytes. So they are having the nucleus in different shapes and structure. Then comes the platelets. So platelets are what? Are tiny disc shaped cells that helps in the regulation of the blood flow. Whenever there is any damage in our tissue or any body part, these platelets actually helps in preventing of the blood loss by providing a mesh-like structure over that injured region. So what is the composition or what are the compositions of the blood? So this is the whole blood. So this blood is mainly comprised of two main parts, that is the plasma and the formed elements or the cellular components. So the plasma contains what? Water and some chemical substances like the amino acids, nitrogenous waste, electrolytes, gases, nutrients, proteins. So these proteins are comprised of albumins, fibrinogens, and globulins. So this proteins helps in the mechanism of the immune system and the blood clotting mechanism. And in the formed elements, it comprised of erythrocytes, that is the red blood cells, leukocytes, that is the white blood cells, and the platelets. So these leukocytes are divided into the granulocytes and the agranulocytes. Granulocytes actually comprised of what? Neutrophils, basophils, and eosinophils. And agranulocytes comprised of lymphocytes and monocytes. So what is the function of the blood? As we know, blood actually carries a essential importance in our body. So the first function is the fluid connective tissue. As it is a connective tissue, it is composed of 55% of plasma and 45% of the cellular components, which includes RBCs, WBCs, and platelets. So these living cells are actually suspended in the plasma part of our blood, and this is known as the fluid connective tissue, and not just a fluid, as it contains what? Different types of cells, proteins, and formed elements. That's why it is known as the fluid connective tissue. Then it provides oxygen to the cells. So this blood absorbs oxygen from the lungs and transports it to the different cells of the body. The waste carbon dioxide moves from the blood to the lungs and is exhaled. Then comes the transports of hormones and the nutrients. So the blood, in the case of the endocrine system, what we know that in endocrine system, the hormones actually transfers from the one part of the organ side to the another part of the organ side through the blood, with the help of blood, as it is a ductless gland. So the passage or the transfer or the movement of the hormones actually takes place with the help of the blood only. Then comes the nutrients, that is the digested nutrients such as the glucose, vitamins, minerals, and proteins are absorbed into the blood through the capillaries in the villi, lining the small intestine. So these hormones are secreted by the endocrine glands and are also transported by the blood to the different organs and the tissues. Then comes the homeostasis. Homeostasis means what? To maintain the normal physiology, which includes the internal body temperature or absorbing or releasing of the heat and miscellaneous activities. Normal balance of the body is done by the blood. Then comes the clotting of the blood at the site of the injury. As I have mentioned earlier, that the platelets helps in the clotting mechanism of the blood at any injured site. So this platelets does what? It comes at the surface of the injured site and forms a fibrin-like form of clot at the wound site. So there is a temporary plaque formation or prevents the loss of the blood. Then comes the transport of the waste material to the kidney and the liver. So this blood enters the kidney where it is filtered to remove nitrogenous waste out of the body. Blood plasma and the toxins from the blood are also removed by the liver. Then comes the protection against the pathogens. The WBCs fight against the infections as they are having neutrophils, eosinophils, basophils, monocytes, and lymphocytes. They actually engulf the bacterial or infectious agents and stops the multiplication of the infection in our body. Then what are the types of the blood vessels? So in this diagram we can see there is a red one and there is a blue one, and in between there is a mesh like network like structure. So this red one is denoted as a artery that is carrying a oxygenated blood, and this blue one is the vein that carries the deoxygenated blood. So this artery is having arterioles. So this is the subdivision of the artery which is known as arterioles. So this arterioles is forming a capillary structure. This is the capillary structure. And these capillaries are then attaching with the veins via a structure that is a venule. So this venules is the subdivision structure of the veins. So there is three types of blood vessels: arteries, veins, and capillaries. So arteries are the strong tubes and muscular in nature. These blood vessels actually carries what? Oxygen rich blood, that is oxygenated blood from the heart to the all the tissue of the body. So aorta is the largest artery present in our heart that actually carries the oxygenated blood to the other parts of the body. Then comes the veins. Veins are the elastic blood vessels which carry deoxygenated blood from all parts of the body to the heart. An exception is the umbilical and the pulmonary veins. So the veins are carrying what? The deoxygenated blood from the body to the heart. So from the heart, what happens? It actually goes to the lungs where the carbon dioxide actually exhaled out at the environment, and oxygenated related blood oxygen is added to the blood and it carries to the heart via a pulmonary veins. Then comes the capillaries. On reaching the tissues, the arteries branch further into extremely thin tubes called capillaries. So these capillaries have a very minute pores which actually helps in the passage of the nutrients or the oxygen or the hormones into the other side of the tissue, surrounding tissue present around the capillaries. Then comes the then comes the hemostasis. Hemostasis, as the name hem is related to the blood, right? So hemostasis is what? It is a physiological defensive reaction to an injury or a cut that seals the blood vessels and thus helps in healing. So hemostasis is a physiological process, defensive process, where whenever there is an injury or cut, it actually helps in sealing of that blood vessels to prevent further bleeding. So mainly platelets, endothelial cells of the blood vessels, and blood proteins are responsible for hemostasis. Hemostatic mechanism proceeds in the following series of the steps. Firstly, there is a changes in the blood vessels. Then there is a blood clotting formation. Then forms the platelet plug formation. So this is the mechanism of the hemostasis. So in the mechanism of hemostasis, firstly what happens whenever there is a breakage of blood vessels? So there is a first process which comprises of vasoconstriction, that is a constriction of that broken blood vessels. So what happens after the injury? The platelets actually, so these are the platelets, actually comes in contact with the damaged blood vessels. And this damaged blood vessels, the surface becomes sticky in nature and this stick to the damaged wall. So this platelets actually helps in the release of a chemical neurotransmitter, that is the serotonin, 5-HT, and thromboxans. This two substance actually helps to constrict the blood vessels and stops further any blood flow through it. So the platelet plug formation, then comes the platelet plug formation. So this platelet plug formation occurs when the sticky platelets clump together and releases the ADP, that is adenosine diphosphate, and attract more platelets to the injured site. So more platelets are coming at the site of the injury and sticks with the previous platelet. So this one was the previous platelet and new platelets are also coming and attaches to the previous platelet at the damaged site. and it quickly acts as what? A temporary plug formation. A temporary seal is happening at the second stage. So there is a temporary seal or known as the platelet plug formation. Then comes the development or the coagulation of the clot or the blood clotting factors. In this step, what happens with the help of different clotting factors activation, there is a formation of the prothrombin. This prothrombin activates what? The thrombin. This thrombin then converts the soluble fibrinogen substance to the insoluble fibrin substance. This insoluble fibrin actually comes and forms a mesh-like structure and forms a blood clot. This mesh-like structure or the fibrin strands prevents the blood coming out from the injured site. So this is the general mechanism of the hemostasis. Then comes the clotting factors. So there are several clotting factors, that is the fibrinogen, prothrombin, tissue factor. So first, these clotting factors are actually denoted in a Roman numerical form. So factor number 1 is fibrinogen. Factor number 2 is prothrombin. Factor number 3 is tissue factor or thromboplastin. Factor number 4 is calcium. Factor number 5 is known as labile factor or proacyllerin or AC globulin. Factor number 7 is stable factor or proconvertin. Factor number 8 is antihemophilic globulin or antihemophilic factor A. Factor number 9 is Christmas factor or plasma thromboplastin component or antihemophilic factor B. Factor number 10 is Stewart Power factor. Factor number 11 is plasma thromboplastin antecedent or PTA factor or antihemophilic factor C. Then comes the factor number 12, that is the Hedgeman factor, and factor number 13, the fibrin stabilizing factor. So there is no factor number 6. Vitamin K is also helps in the synthesis of what? Factors 2, factor 7, factor 9, and factor 10. So the blood clotting mechanism follows two systems or two pathways, that is the intrinsic system and the extrinsic system pathway. So this intrinsic system is actually occurs when the injury is happened inside the body or the collagen fibers, or there is a change in the blood constituent. And extrinsic pathway follows when there is an injury outside the blood vessels wall or other body tissues. So what happens in the intrinsic system pathway? The first factor is the factor number 12, that is the Hedgeman factor, right? So this inactive Hedgeman factor gets activated due to the change in the blood constituent or the exposure of any collagen fibers. So this inactive 12 Hedgeman factors is activated into the activated 12 factor. This activated 12 factor actually activates the factor number 11 into a factor number 11 factor. This factor number 11 activates the factor number 9, that is the Christmas factor, into an active form. Then what happens? The antihemophilic factor, factor number 8, actually initiates the activation of Stewart Power factor, that is factor number 10. So then comes the extrinsic system. So this extrinsic system occurs when there is a damage in blood vessel wall, and this causes the release of the tissue thromboplastin factor number 3. See, this factor number 3 actually activates the factor number 7 from the inactive form. So this 7 factor then activates the factor number 10 factor, that is the Stewart Power factor. So one thing it is common, we can see that the intrinsic pathway and extrinsic pathway actually coming in contact with a common pathway system, that is activation of the factor number 10 is happening in both the system, right? So there is a inactive form of 10 which is activated by the factor number 9 in case of the intrinsic system, and in extrinsic system the inactive factor number 10 is activated by the factor number 7. With respect with the other subconstituent of the blood clotting factor, that is the calcium, platelet phospholipids, and factor number 3. So when this common reaction is helps to activate the factor number 10, this factor number 10 actually does what? It actually activates the factor number 2 to convert them into the thrombin. So basically thrombin is not present in our body in an active state. So what happens? This factor number 10 actually helps in the conversion of the prothrombin into a thrombin compound. Then this thrombin does what? With the help of the factor number 13, that is the fibrin stabilizing factor, along with the calcium, it actually converts the soluble fibrinogen factor number 1 to the insoluble fibrin structure. This fibrin actually forms a clot or mesh like or thread like structure after 5 to 30 minutes. So this is the mechanism or the pathway, two pathway, that is intrinsic system pathway and extrinsic system pathway of the blood clotting. Then comes the blood groups. So blood groups. So blood groups are invented or discovered by a pathologist or immunologist. He was an Austrian American immunologist, and his name was Karl Landsteiner. In 1901, he discovered the human blood group system. So Karl Landsteiner helps to determine what the blood groups. So it actually helps in what the study of blood transfusion reaction. He was awarded Nobel Prize for his discovery in the physiology or medicine in 1930 for this discovery. So why we need to determine the blood groups? So in earlier, the patient actually died before 1900 due to the blood transfusion reaction. That is, different blood group people are receiving different blood group from different types of people, and this difference in the blood group is happening what? A blood transfusion reaction. So for this we need to know who is having what type of blood group, and that person should be given what type of blood group, or that person will give what type of blood group to which person, right? So there is a discovery of blood group, that is ABO group. Further, in the 1902, the fourth main group was founded by the Dacasterello and Sturley, that is the AB blood group. And then in 1940, by Landsteiner and Wiener, they actually discovered the rhesus system, that is the positive and the negative determination of that particular blood group. So this is a common general blood group type, four types of blood group, right? So this column is a blood group A, B, AB, and O. This represent the antigen and antibody and donor and recipient. So what happens to the A blood group type? So A blood group type person is having RBCs, that is the red blood cells. So this red blood cells at the around the surface of the red blood cells of the A blood group type, they are having A type of antigen, right? So this A type of antigen, as they are having A type of antigen, that particular person actually produces the opposite type of antibody, not the same type of antibody. If he or she produces the same type of antibody which doesn't occur in normal human physiology, then what happens? The antigen antibody will react and it can cause a blood transfusion reaction. So mainly what happens in our human normal physiology, a particular blood group type having a particular antigen doesn't produce the same type of antibody. It actually produces a different type of antibody. So in the blood group A type, the A red blood cells are having A type of antigen. So it will produce or produces what? The antibody B. If it is having antigen A, then it will produce antibody B. Right? So donors. So who can be the donors and who can be the recipients? So the A type of blood group can give blood to A type of blood group people and AB type of blood group people. That you will know at the further explanation why A and AB are considered to be compatible. And incompatible are B and O. So A blood group type cannot give the blood to B and O type of blood group because both the B and O type of blood group actually makes what? A type of antibody and A type of antigens can react with the A type of antibody. So B and O actually produces what? A type of antibody. That A type of antibody can react with the A type of antigen. Right? And as a recipient, from whom he or she can take the blood? From the A and O. But it will be incompatible with the B and AB. Why? Because A makes the antibody B and that will react with the antigen B. B antigen. Then comes the B blood group type. So in the B blood group type, similarly, the RBCs are having B antigen, and in respect to the B antigen, that body will create what? Antibody A. Not that same type of antigen, but the different type of antibody. So it will be compatible to be donor as B and AB. They can give blood to B and AB, but they will not give blood to the A and O. Why? Because A and O both can make the B antibody. So what happens? That B antigen of the B blood group type can react with the B antibody of the A and O type of person. And next is the recipient. So from whom he can take the blood? From the B and O, but not from the A and AB. Why? Because the B blood group type can make the antibody A type of antibodies, and that A type of antibodies will react with the A type of antigen which is present in the A type of blood group and AB type of blood group. Then comes the AB blood group. AB blood group actually carries both type of antigen, that is antigen A and antigen B, to their surface of the RBC, but they are not having any antibody or antibody A or antibody B production. So to whom it can give the blood? It can give the blood to the AB type only. So the AB type blood group can only give the blood to AB type person. Why? Because other blood group, that is AB, A, B and O, these three type of blood groups all make the antibodies to that the respect of the antigen A and B that is present in the AB antigens. And to from whom it can take the blood? From the universal recipient, compatible with all groups, that is universal recipient. AB makes no antibodies, so it can receive from any blood group type, as AB doesn't make any antibodies. So if A blood group type is giving blood to the AB, then it will not react because AB blood group type person never makes any antibodies. Similarly, B and O, if they give the blood to the AB type, it will not react as there is no antibody production in case of AB blood group type person. Then comes the O blood group type. So O blood group type person neither have A nor B antigen in their RBCs, but they produces antibody A and antibody B. So from whom it can take? It can give the blood. It is a universal donor. O is always considered to be universal donor. Why? O red blood, as it doesn't have any antigen. As it doesn't have any antigen, so if we can give to any blood group type, it will not stimulate any antibody A or antibody B to the respective other blood groups. Then comes from whom it can take the blood. It can only take the blood from the O type. Why? As A, B, and B type of makes the A and AB and B, because type O makes what? The antibody A and antibody B. So the O type of blood group person is having no antigen, but they are having antibody A and antibody B. If suppose any person is giving other group blood group type person, that is AB, A and B, gives the blood to the O type, then their antigen can react with the antibodies of A and AB of O blood group type. So this is only be compatible with the O only. So in the blood group section, as we have known, that the universal recipient is AB and universal donor is O. Then comes the most important part, that is the rhesus system, right? So the rhesus system was first discovered in which animals? The rhesus monkey. And it is also known as the RH system. So this rhesus system consists of 6 antigens, but they actually doesn't produce any antibodies in general. So this out of this 6 antigen, most significant and prominent antigen immunologically is estimated to be D antigen. So this Rh system has two blood groups, Rh positive, Rh negative. So whenever a person is having a Rh positive, it is considered that the person is having a D antigen present over the RBCs. And vice versa, if it is having a Rh negative, then there is no D antigen present in the RBC. So in general, in the Indian population, it is considered that 95 to 98% of the populations are having Rh positive and 22 to 5% are having Rh negative. So what happened? A person with Rh blood doesn't have any Rh antibodies naturally in the blood cell. But a person with Rh negative blood can develop the Rh antibodies in the blood plasma if he or she receives blood from a person with Rh positive blood. So what happens? Normally Rh negative blood does not have any Rh antibodies. But whenever any Rh negative blood person receives Rh positive blood, then that Rh negative blood person can develop the Rh antibody against that Rh positive blood, whose Rh antigens can trigger the production of Rh antibodies. And this can create a problem and causes a blood transfusion reaction. So, what is the principle behind the estimation of the blood group type test? So, it follows as one principle, that is the agglutination reaction or the blood clumping. So, what do you mean by agglutination reaction? Agglutination reaction means is what? When there is a reaction between agglutinogens and agglutinins. So, what is agglutinogens? Agglutinogens means the antigens and agglutinin means the antibodies. So whenever there is a reaction between the agglutinogens and agglutinogens and agglutinins, it causes what? The blood clumping or hemolysis of the RBCs, which is not good for our normal health or the physiology. So what happens for the testing of the blood group in the lab? We need a monoclonal antibody which can mimic our immune system ability to fight over the pathogens, right? So the monoclonal antibodies in the lab are available in three types: A, B, and Rh antigens for the this type of antigen. So monoclonal antibody acts against the antigen A, also known as antibody A, which is comes in a small bottle with a dropper, and it is denoted with the universal color, that is the blue color. Then comes the antibody B for antigen A. It comes in a yellow color solution, and then comes the antibody D for the antigen Rh antigen. So it is generally a colorless solution. So this monoclonal antibody, artificial antibodies, actually helps to mimic the immune system of a normal physiological human being, and it should be kept in a normal in a refrigerator to be stored in, and it is recommended to tilt the bottle a couple of times before use to order in to resuspend the antibodies that have settled down at the bottom of the bottle. So all these colors are universal in nature, right? So, what is the procedure for that blood grouping test? So, the patient or the individual should sterile their finger with the alcohol. Then, with the help of a lancet, the finger will be pricked, and it should be allowed, and the blood should be allowed to come out. So, we will make a four well, or the four area, where each four area will contain a four drops of blood, like this way. Then we will add antibody A to the first place of the blood, then antibody B to the second place of the blood, and antibody D to the third place, and this fourth place is control for determination of the is if the taste is accurate or not, right? So what happens after giving the antibody A, B, and D? In the first case, as we can see, for the O positive, there is no agglutination reaction forms in the antibody A, antibody B, only anti D. So what we can estimate, as we have read in previous slide, that O group blood group type person does not have any A or B antigen, right? But it produces AB antibody. So as our blood is having no antigen in case of O positive O person, so we are giving the antibody A and antibody B, and thus there is no reaction. As there is no antigen, thus we can estimate that this section of the test is considered to be the O blood group type. And as there is an agglutination form in the antibody D, we can conclude that there is agglutination reaction, means what? This type of person is having Rh antigen. That's why that Rh antigen is reacting with the Rh antibody, and thus it is creating what? The agglutination reaction. So this is the O positive blood group type. Similarly, if clumping of RBCs in antiserum A occurs, suppose this is the antiserum A, then it is the A blood group type. Positive, if positive, then agglutination will happen in D also. If negative, A will agglutinate, but D will not agglutinate. Similarly, in the B blood group type, if the agglutination occurs in the B well and the D well, it will be positive. If only B, then it will be negative. And if the agglutination happens in both the anti-A, anti-B, then it will consider to be the AB positive. Why? As AB is having both antigen and anti-B, and that both the A and B antigen is reacting with both A antibody and A B antibody. And for the positive part, the D antigen is present. That's why it is reacting with the D antibody. That's why it is the AB positive. Vice versa, there is the AB negative. And this one is the invalid. Due to some reason, the result came invalid. So what is the clinical significance of the blood group determination? So, firstly is the blood transfusion. So, in the blood transfusion case, the blood group of the recipient and donor must be matched. Otherwise, agglutination or clumping of the RBC occurs. And this will result in what? Fetal reaction. And clumping of the RBCs or the fetal death can also occur. Then comes the hemolytic disease of the newborn and erythroblastosis fetalis. Then comes the testing of the paternity disease of the newborn, also in the medico-legal practice of the paternity disputes. Then comes the most important part, that is the erythroblastosis fetalis. So what happens in the erythroblastosis fetalis? In the erythroblastosis fetalis, suppose the father is having Rh positive, that is antigen is present, right? And mother is having Rh negative. So mother is having negative blood group and father is having positive blood group. And when this mother is conceiving, their child is actually inheriting the blood group type from the father, that is positive blood group. As it is a dominant allele, that's why that fetus will inherit the dominant type of blood group from the father, that is Rh positive. So what happens during the first pregnancy? So mother is having Rh negative in her blood, but the baby is carrying Rh positive blood in his body. So what I have discussed in the previous slide, that Rh negative person actually doesn't have any antibody. But if they receive any Rh positive blood, then their immune system can trigger to produce the antibody against that positive antigen, blood group type antigen. So what happens in here, this positive blood group will pass through the placenta and will trigger the maternal lymphocyte. This maternal lymphocyte will create antibodies against that positive Rh blood group. And that positive Rh blood group antibody will cross the placenta and will attack the immune system or the baby's red blood cells. So this fetal red blood cell actually damages or hemolysis occurs. So then comes another theory that erythroblastosis fetalis always does not affect the baby fetus in during the first pregnancy, as the concentration of the antibody is low. But during the second pregnancy, what happens? The mother's blood is already having a antibody against the positive blood group, right? So again, what happens? In the second child is also having carrying the positive blood group from the father, and as the mother is already having a antibody for the Rh positive from the previous pregnancy, that will initiate and accumulate more antibodies for the Rh positive antigen blood group, and that will attack the baby and will cause fetal damage by clumping of red blood cells. So this can be prevented by applying or administering a medicine, that is a ROGAM. So this ROGAM can prevent the formation of the lymphocytes that can trigger or develop the antibody against that Rh positive antigen that is having inside the baby. So this is the erythroblastosis fetalis. So these are the references, and thank you for listening.